In this video, I will show you how to use the Daniell code DCC to find out about the regulatory regions around a specific gene. We will use the gene MACE2A as an example. In order to do this, we first go to the UCSC genome browser with our Daniell code tracker loaded, which is easiest done via the navbar on the top here. When you are at the genome browser, go to the gene of interest with the search field. So we type in MACE2A here and select it. We will use the PADRES annotation track to find out about the regulatory regions in our gene. PADRES is a combination of Chrome-HMM classification and open chromatin regions. It can be found in the Stages Types section of the tracks down here. Make sure that only PADRES is selected, and we'll show them as squish to make it more compact. You see now above the RefSec and the GAP track, 5 PADRE tracks loaded, ordered by stages. And we'll see here, in one of these intronic regions, that the later stages suddenly these red blocks appear. And if we hover over them, we can see that these are all enhance elements. This means, besides being open chromatin regions according to ATAC-seq, they also have a strong H3K27 acetylation and H3K4 monomethylation signal. Let's zoom in and let's look for cell type specificity. For that, we use the cell type annotation track, which is based on single-cell ATAC-seq data. Go to the cell types page and load show them all in full display mode. And it turns out that our enhancer regions are only open in CNS samples, indicating that we have here a CNS-specific enhancer candidate. With that, I hope I could give you some ideas on how to use the Daniell code DCC.